Carbohydrate metabolism refers to the set of biochemical processes that manage the breakdown and synthesis of carbohydrates in the body, which serves as a primary energy source. It involves several key pathways. 1. Glycolysis. Location: cytoplasm of the cell. Process: breaks down glucose into pyruvate, producing ATP and NADH. Outcome: for each molecule of glucose, glycolysis generates 2 ATP molecules and 2 NADH molecules. 2. Glycogenesis. Location: liver and muscle cells. Process: converts glucose into glycogen for storage. Outcome: this process helps in storing excess glucose as glycogen for later use. 3. Glycogenolysis. Location: liver and muscle cells. Process: breaks down glycogen into glucose 1-phosphate, which is then converted to glucose 6-phosphate. Outcome: it helps maintain blood glucose levels during fasting or between meals. 4. Gluconeogenesis. Location: liver, mainly, and kidneys. Process: synthesizes glucose from non-carbohydrate sources like amino acids, lactate, and glycerol. Outcome: maintains blood glucose levels during prolonged fasting or intense exercise. Number 5. Citric acid cycle, Krebs cycle. Location: mitochondria. Process: pyruvate from glycolysis is converted to acetyl-CoA, which enters the Krebs cycle to produce NADH, FAH2, and ATP. Outcome: it is central to energy production, providing electrons for the electron transport chain. 6. Pentose phosphate pathway, PPP. Location: cytoplasm. Process: converts glucose 6-phosphate into ribose 5-phosphate, used for nucleotide synthesis, and produces NADPH. Outcome: NADPH generated is crucial for biosynthetic reactions and maintaining cellular redox balance. 7. Oxidative phosphorylation, electron transport chain. Location: mitochondrial inner membrane. Process: uses NADH and FADH2 from glycolysis, the Krebs cycle, and the PPP to produce ATP. Outcome: this process is the major source of ATP in aerobic organisms. Regulation and hormonal control. Carbohydrate metabolism is regulated by hormones such as insulin, lowers blood glucose by promoting glycogenesis and glycolysis. Glucagon raises blood glucose by promoting glycogenolysis and gluconeogenesis. Cortisol and epinephrine also help in raising blood glucose levels during stress or low blood sugar conditions. Together, these processes ensure the balance between energy supply and demand, adapting to different metabolic needs during feeding, fasting, and stress. Glycolysis is a fundamental metabolic pathway that breaks down glucose, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon compound. This process occurs in the cytoplasm of cells and is anaerobic, does not require oxygen. Glycolysis is essential for energy production as it generates ATP and provides intermediates for other metabolic pathways. Overview of glycolysis. Location: cytoplasm. Net energy yield: 2 ATP and 2 NADH per glucose molecule. Overall reaction: glucose plus 2 ATEX squared plus 2 ADP plus 2 text PI, right arrow 2 text pyruvate plus 2 NADH plus 2 HX2 plus 2P AT text H2 text O. Phases of glycolysis. Glycolysis can be divided into 2 main phases. 1. Preparatory phase, investment phase. Purpose: converts glucose into 2 molecules of glyceraldehyde 3-phosphate, G3P. This phase requires an input of 2 ATP. Steps: 1. Phosphorylation of glucose. Glucose is phosphorylated by hexokinase or glucokinase in the liver to form glucose 6-phosphate, G6P. This step consumes 1 ATP. 2. Isomerization. Glucose 6-phosphate is converted to fructose 6-phosphate, F6P, by the enzyme phosphoglucose isomerase. 3. Second phosphorylation. Fructose 6-phosphate is phosphorylated by phosphofructokinase 1, PFK1, to form fructose 1,6-bisphosphate, F16BP. This step consumes another ATP and is a key regulatory point in glycolysis. 4. Cleavage. Fructose 1,6-bisphosphate is cleaved by aldolase into 2 3-carbon molecules, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. 5. Isomerization of DHAP. DHAP is converted into G3P by the enzyme triose phosphate isomerase, resulting in 2 G3P molecules. 2. Payoff phase, energy generation phase. Purpose: produces ATP and NADH while converting G3P into pyruvate. Steps: 1. Oxidation and phosphorylation. Glyceraldehyde 3-phosphate is oxidized by glyceraldehyde 3-phosphate dehydrogenase, producing 1,3-bisphosphoglycerate. 1,3-BPG, and NADH. 2. ATP production, substrate-level phosphorylation. 1.3-BPG transfers a phosphate group to ADP, forming 3-phosphoglycerate, 3-PG, and producing 1 ATP per G3P, total of 2 ATP per glucose. 3. Conversion to 2-phosphoglycerate. 3-phosphoglycerate is converted to 2-phosphoglycerate, 2,2-PG, by phosphoglycerate mutase. 4. Dehydration. 2-phosphoglycerate is dehydrated by enolase to form phosphoenol pyruvate, PEP. 5. Second ATP production. PEP transfers a phosphate group to ADP, forming pyruvate and generating another ATP, total of 2 ATP per glucose. Energy yield of glycolysis. Glycolysis consumes 2 ATP in the preparatory phase but produces 4 ATP in the payoff phase, leading to a net gain of 2 ATP per molecule of glucose. NADH. 2 molecules of NADH are produced, which can later be used in oxidative phosphorylation if oxygen is present to generate more ATP. Regulation of glycolysis. Glycolysis is tightly regulated at 3 key enzyme-catalyzed steps. 1. Hexokinase, glucokinase, step 1, inhibited by its product, glucose 6-phosphate in many tissues. Glucokinase in the liver has a higher Km, allowing it to handle high glucose levels. 2. Phosphofructokinase 1, PFK1, step 3. This is the rate-limiting step of glycolysis. It is activated by AM, signifying low energy, and fructose 2,6-bisphosphate, and inhibited by ATP and citrate. 3. Pyruvate kinase, step 10, inhibited by ATP and alanine, and activated by fructose 1,6-bisphosphate, feed-forward activation. Fate of pyruvate. After glycolysis, the fate of pyruvate depends on the presence or absence of oxygen. Aerobic conditions. Pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase, which then enters the citric acid cycle, Krebs cycle, for further oxidation. Anaerobic conditions. Pyruvate is converted into lactate by lactate dehydrogenase in animal cells, e.g., in muscles during intense exercise. In yeast and some bacteria, pyruvate undergoes fermentation to produce ethanol and CO2. Importance of glycolysis. Energy production. Provides quick ATP, especially important in cells with high energy demands like muscle cells. Intermediate production. Produces intermediates like 3-phosphoglycerate and pyruvate, which can enter other metabolic pathways such as gluconeogenesis and amino acid synthesis. Since red blood cells lack mitochondria, glycolysis is their sole source of ATP. Glycolysis serves as a versatile metabolic pathway that connects to many other processes in the body, making it crucial for maintaining cellular energy homeostasis. Pyruvate metabolism is a key metabolic junction, as it can be directed toward multiple pathways depending on the cell's needs and the availability of oxygen. The fate of pyruvate determines whether it will produce energy, serve as a substrate for biosynthesis, or contribute to the body's balance of glucose and other metabolites. Overview of pyruvate metabolism. Pyruvate is a 3-carbon compound that results from glycolysis. It can be metabolized into various products based on cellular conditions, aerobic or anaerobic, and the specific needs of the tissue. Pathways of pyruvate metabolism. 1. Conversion to acetyl-CoA, aerobic conditions. 2. Lactate fermentation, anaerobic conditions. 3. Conversion to oxaloacetate. 4. Conversion to alanine, transamination. 5. Ethanol fermentation, in yeast. 1. Conversion to acetyl-CoA. Location, mitochondrial matrix. Enzyme, pyruvate dehydrogenase complex, PDC. Reaction, pyruvate plus NADMI plus CoA, acetyl-CoA plus CO2 NADH. Outcome, acetyl-CoA enters the citric acid cycle, Krebs cycle, for further oxidation, which produces more NADH, FADH2, and ATP via the electron transport chain. Regulation, activated by pyruvate and NADMI. Inhibited by high levels of ATP, acetyl-CoA, and NADH, indicating sufficient energy levels. Importance. This is the primary pathway for energy production from pyruvate in cells with sufficient oxygen supply. It links glycolysis with the citric acid cycle and ultimately leads to ATP production through oxidative phosphorylation. 2. Lactate fermentation. Location, cytoplasm, especially in muscle cells, red blood cells, and other anaerobic conditions. Enzyme, lactate dehydrogenase, LDH. Reaction, pyruvate plus NADH, lactate plus NADMI. Outcome, regenerates NADMI, allowing glycolysis to continue in the absence of oxygen. Importance, allows ATP production through glycolysis when oxygen is scarce, e.g., during intense exercise. In muscle cells, lactate can be transported to the liver where it is converted back to glucose through gluconeogenesis, Cori cycle. Accumulation of lactate can cause muscle fatigue and soreness due to the decrease in pH. 3. Conversion to oxaloacetate. Location: mitochondria and cytoplasm for gluconeogenesis. Enzyme pyruvate carboxylase. Reaction: pyruvate plus CO2 plus ATP, oxaloacetate plus ADP plus pi. Outcome: oxaloacetate can enter the citric acid cycle as an intermediate. It is also a critical intermediate for gluconeogenesis in the liver and kidneys. Importance: this pathway is crucial for maintaining the levels of citric acid cycle intermediates, anaplerotic reaction. It is also essential for glucose production from non-carbohydrate sources, helping maintain blood glucose levels during fasting. 4. Conversion to alanine, transamination. Location: cytoplasm. Enzyme: alanine transaminase, ALT. Reaction: pyruvate plus glutamate, alanine plus A-ketoglutarate. Outcome: converts pyruvate into alanine, which can be transported to the liver. Importance: part of the glucose-alanine cycle, where alanine transports amino groups from muscle to the liver. In the liver, alanine can be converted back to pyruvate, which can then be used for gluconeogenesis, while the amino group is used for urea synthesis. This helps in maintaining muscle mass during prolonged fasting by allowing muscle tissue to provide substrates for gluconeogenesis without breaking down too much muscle protein. 5. Ethanol fermentation in yeast and certain bacteria. Location: cytoplasm of yeast and some anaerobic bacteria. Enzymes: pyruvate decarboxylase and alcohol dehydrogenase. Reaction: pyruvate, acetaldehyde plus CO2, acetaldehyde NADH, ethanol plus NADMI. Outcome: regenerates NADMI, allowing glycolysis to continue under anaerobic conditions. Importance: this pathway is used in the production of alcoholic beverages and in the baking industry. It is not significant in human metabolism, but is an important aspect of microbial energy production under anaerobic conditions. Summary of pyruvate metabolism. Paths. Conditions: main product, outcome. That is an important aspect of microbial energy production under anaerobic formation. Acetyl-CoA formation. Aerobic: acetyl-CoA, pyruvate dehydrogenase, enters citric acid cycle for further energy production. Lactate fermentation, anaerobic. Lactate dehydrogenase, regenerates NADMI for continued glycolysis. Oxaloacetate formation, gluconeogenesis, oxaloacetate, pyruvate carboxylase, enters gluconeogenesis or citric acid cycle. Alanine formation, amino acid metabolism. Alanine transaminase, transports nitrogen to the liver. Ethanol fermentation, anaerobic yeast, ethanol plus CO2, pyruvate decarboxylase, alcohol dehydrogenase, used in fermentation by yeast. Regulation of pyruvate metabolism. Pyruvate metabolism is tightly regulated to match the cell's energy demands and availability of substrates. High ATP, high NADH directs pyruvate away from energy production, favoring storage pathways, e.g. gluconeogenesis. Low ATP, high ADP promotes conversion of pyruvate to acetyl-CoA for the citric acid cycle. High acetyl-CoA inhibits pyruvate dehydrogenase, promoting pyruvate carboxylase activity, which channels pyruvate toward gluconeogenesis. Importance of pyruvate metabolism. Energy production. Provides a critical link between glycolysis and the citric acid cycle, determining the cell's energy output. Gluconeogenesis. Maintains blood glucose levels during fasting. Amino acid synthesis. Supplies substrates for the synthesis of alanine and other amino acids. Anaerobic ATP production. Allows cells to generate ATP when oxygen is limited. Pyruvate metabolism is central to cellular energy homeostasis, acting as a bridge between various metabolic pathways and adapting to changing conditions within the cell and the body. The Krebs cycle, also known as the citric acid cycle, CAC, or tricarboxylic acid, TCA cycle, is a crucial part of cellular respiration. It takes place in the mitochondrial matrix and serves as a central hub for the metabolism of carbohydrates, fats, and proteins. The primary role of the Krebs cycle is to oxidize acetyl-CoA derived from pyruvate or other sources into carbon dioxide, CO2, while capturing high-energy electrons in the form of NADH and FADH2. These electron carriers are used in the electron transport chain, ETC, to produce ATP through oxidative phosphorylation. Overview of the Krebs cycle. Location, mitochondrial matrix. Initial substrate, acetyl-CoA. Final products per cycle turn, 2 CO2, 3 NADH, 1 FADH2, and 1 GP or ATP. Net ATP yield? Approximately 10 ATP per turn when coupled with oxidative phosphorylation via NADH and FADH2 oxidation. Steps of the Krebs cycle. The Krebs cycle consists of 8 steps that regenerate the starting compound oxaloacetate with each turn of the cycle. Below is a detailed breakdown. 1. Formation of citrate. Reaction: acetyl-CoA, 2 carbons, plus oxaloacetate, 4 carbons. Citrate, 6 carbons. Enzyme: citrate synthase. Importance: this step is highly exergonic, releases energy, and is regulated to control the flow of the cycle. 2. Isomerization of citrate to isocitrate. Reaction: citrate, isocitrate. Enzyme: aconitase. Mechanism: this involves a dehydration followed by a rehydration, forming isocitrate. This rearrangement is necessary to make the molecule more reactive for the next step. 3. Oxidation of isocitrate to aketoglutarate. Reaction: isocitrate NADMI aketoglutarate plus CO2 plus NADH. Enzyme: isocitrate dehydrogenase. Importance: this is the first of 2 decarboxylation reactions in the cycle, and it is a key regulatory step. The reaction releases 1 molecule of CO2 and produces NADH. 4. Oxidation of aketoglutarate to succinyl-CoA. Reaction: aketoglutarate CoA plus NADMI, succinyl-CoA plus CO2 plus NADH. Enzyme: alpha-ketoglutarate dehydrogenase complex. Importance: this step is similar to the pyruvate dehydrogenase reaction and involves the decarboxylation of aketoglutarate, forming a high-energy thioester bond in succinyl-CoA. It generates another molecule of NADH. Conversion of succinyl-CoA to succinate. Reaction: succinyl-CoA plus GDP or ADP plus pi, succinate plus GTP or ATP plus CoA. Enzyme: succinyl-CoA synthetase. Importance: this is the only step in the Krebs cycle that directly generates a high-energy phosphate bond, GTP or ATP, through substrate-level phosphorylation. 6. Oxidation of succinate to fumarate. Reaction: succinate plus FAD, fumarate plus FADH2. Enzyme: succinate dehydrogenase. Location: unlike other enzymes of the Krebs cycle, succinate dehydrogenase is embedded in the inner mitochondrial membrane and also functions as COC complex 2 in the electron transport chain. Importance: FADH2 generated here donates electrons directly to the ETC, resulting in approximately 1.5 ATP per FADH2. 7. Hydration of fumarate to malate. Reaction: fumarate plus H2O, malate. Enzyme: fumarase, fumarate hydratase. Mechanism: this step adds a water molecule across the double bond of fumarate to form malate. 8. Oxidation of malate to oxaloacetate. Reaction: malate plus oxaloacetate NADH. Enzyme: malate dehydrogenase. Importance: this final step regenerates oxaloacetate, allowing the cycle to continue, and produces the third NADH of the cycle. Summary of energy yield from the Krebs cycle. Each turn of the Krebs cycle results in 3 NADH. Each NADH yields about 2.5 ATP during oxidative phosphorylation. 1 FADH2. Each FADH2 yields about 1.5 ATP during oxidative phosphorylation. 1 GTP, or ATP, produced directly through substrate-level phosphorylation. 2 CO2, released as a waste product. Net yield per turn of the cycle, approximately 10 AT, 3 NADH, 2.5 ATP, plus 1 FH, 1.5 ATP, plus 1 GTP. Since 1 molecule of glucose yields to 2 molecules of pyruvate, and each pyruvate generates 1 acetyl-CoA, the complete oxidation of 1 glucose molecule through the Krebs cycle, 2 turns, yields 6 NADH, 2 FADH2, 2 GTP, or ATP, 4 CO2. Regulation of the Krebs cycle. The Krebs cycle is regulated primarily at 3 steps, all of which involve reactions with large negative free energy changes. 1. Citrate synthase. Inhibited by high levels of ATP, NADH, and citrate, feedback inhibition. 2. Isocitrate dehydrogenase, activated by ADP and NADMIDs, which signal low energy status, inhibited by high levels of ATP and NADH. 3. Alpha-ketoglutarate dehydrogenase, inhibited by its products, succinyl-CoA and NADH, and by high levels of ATP. These regulatory mechanisms ensure that the cycle runs efficiently when energy is needed and slows down when ATP is abundant. Importance of the Krebs cycle. 1. Energy production. It is central to aerobic energy production, providing high-energy electron carriers, NADH and FADH2, for the electron transport chain. 2. Biosynthetic intermediates. Provides intermediates like A-ketoglutarate, succinyl-CoA, and oxaloacetate that are used in the synthesis of amino acids, heme, nucleotides, and glucose via gluconeogenesis. 3. Metabolic integration. Connects with glycolysis, fatty acid oxidation, and amino acid metabolism, playing a key role in cellular metabolism. Clinical relevance. Genetic defects. Mutations in enzymes of the Krebs cycle, like fumarase deficiency, can lead to metabolic disorders. Cancer metabolism. Some cancer cells exhibit altered metabolism, Warburg effect, where they rely more on glycolysis than oxidative phosphorylation, even in the presence of oxygen. Mitochondrial diseases. Dysfunction in mitochondrial components of the Krebs cycle can impair ATP production, affecting tissues with high energy demands, such as the brain, muscles, and heart. The Krebs cycle is vital not just for energy generation, but also for the production of various metabolites needed for anabolic processes, making it a central metabolic pathway in cellular physiology. Oxidative phosphorylation is the process by which cells produce ATP, the primary energy currency of the cell, through a combination of electron transport and a proton gradient. This process takes place in the inner mitochondrial membrane and is the final stage of aerobic respiration. It couples the energy released from the electron transport chain, ETC, with ATP synthesis through the ATP synthase enzyme. Overview of oxidative phosphorylation. Location, inner mitochondrial membrane. Key components, electron transport chain, ETC, a series of protein complexes, complexes of 4, and mobile electron carriers. ATP synthase uses the proton gradient generated by the ETC to synthesize ATP. Inputs, NADH2O2 and ADP plus pi. Outputs, ATP, H2O, and oxidized forms of NAD bi and FAD. Components of the electron transport chain, ETC. The ETC is composed of 4 protein complexes, complexes 1, 4, and 2 mobile carriers, ubiquinone, also known as coenzyme Q, and cytochrome C. 1. Complex 1, NADH ubiquinone oxidoreductase. Function, transfers electrons from NADH to ubiquinone, coenzyme Q, forming ubiquinol, QH2. Mechanism. As electrons are transferred, protons, HPs, are pumped from the mitochondrial matrix to the intermembrane space. Result, 4 protons pumped per NADH oxidized. 2. Complex 2, succinate dehydrogenase. Function, transfers electrons from FADH2, produced in the Krebs cycle, to ubiquinone, forming QH2. Mechanism, unlike complex 1, complex 2 does not pump protons across the membrane. Result, contributes electrons to the ETC without directly contributing to the proton gradient. 3. Complex 3, cytochrome BC1 complex. Function, transfers electrons from ubiquinol, QH2, to cytochrome cis, a mobile electron carrier in the intermembrane space. Mechanism, the Q cycle at complex 3 results in the pumping of 4 protons into the intermembrane space for every pair of electrons transferred. Result, contributes to the proton gradient, enhancing the electrochemical potential. 4. Complex 4, cytochrome C oxidase. Function, transfers electrons from cytochrome C to molecular oxygen, O2, the final electron acceptor, forming hot water, H2O. Mechanism, for every molecule of O2 reduced, 2 protons are pumped into the intermembrane space, and 2 more protons combine with O2 to form H2O. Result, the reduction of O2 to H2O is a critical step that prevents the buildup of reactive oxygen species, ROS. 5. Mobile electron carriers, ubiquinone, coenzyme Q, shuttles electrons between complexes 1, 2, and 3. Cytochrome C, a small protein that transfers electrons from complex 3 to complex 4 in the intermembrane space. The proton gradient and chemiosmotic theory. Proton gradient. The electron transport through the ETC generates a proton gradient, proton motive force, across the inner mitochondrial membrane. Protons are pumped from the mitochondrial matrix into the intermembrane space, creating electrical gradient. Positive charge accumulates in the intermembrane space. pH gradient, lower pH, higher H- concentration in the intermembrane space compared to the matrix. Chemiosmotic theory, proposed by Peter Mitchell, it explains that the energy stored in this proton gradient, electrochemical gradient, is used to synthesize ATP. ATP synthesis via ATP synthase, enzyme, ATP synthase, also known as complex Vi. Structure. ATP synthase has two main components, FCO, an ion channel embedded in the inner membrane that allows protons to flow back into the mitochondrial matrix. F1, a catalytic unit that synthesizes ATP from ADP and pi, inorganic phosphate. Mechanism. Proton flow. As protons flow down their gradient through the FCO component, they cause the rotation of a part of the ATP synthase complex. Mechanical energy to chemical energy. The rotation leads to conformational changes in the F1 subunit, enabling it to convert ADP and pi into ATP. Result: about 3 protons need to pass through ATP synthase to generate 1 molecule of ATP. ATP yield in oxidative phosphorylation. The energy yield of oxidative phosphorylation depends on the number of protons pumped and the efficiency of the ATP synthase. Per NADH, contributes to the pumping of about 10 protons across the membrane, 4 by complex 1, 4 by complex 3, 2 by complex 4, yields approximately 2.5 ATP molecules. Per FADH2, contributes to the pumping of about 6 protons, only 4 by complex 3 and 2 by complex 4, as complex 2 does not pump protons. Yields approximately 1.5 ATP molecules. Since the oxidation of one glucose molecule via glycolysis, the Krebs cycle, and oxidative phosphorylation yields 10 NADH, 2 from glycolysis, 2 from the conversion of pyruvate to acetyl-CoA, and 6 from the Krebs cycle. 2H2 from the Krebs cycle. Total ATP yield from oxidative phosphorylation per glucose is approximately 26 to 28 ATP. 2 NADH from glycolysis yields slightly less ATP when transported into mitochondria. Regulation of oxidative phosphorylation. Oxidative phosphorylation is regulated by the availability of ADP. High ADP levels indicate energy demand, stimulating the ETC and ATP synthesis. O2. Oxygen is essential as the final electron acceptor. Without it, electron flow halts, stopping the entire chain. NADH-FADH2, substrate availability from the Krebs cycle is crucial for maintaining the flow of electrons through the ETC. Reactive oxygen species, ROS in the ETC. ROS formation. Incomplete reduction of oxygen can lead to the formation of superoxide anion, O2-Ms, and other reactive oxygen species. Complexes 1 and 3 are the primary sites where ROS can be generated if electron flow is inefficient. Importance of ROS. While ROS can damage proteins, lipids, and DNA, they also serve as signaling molecules at low levels. Cells have antioxidant systems like superoxide dismutase, SOD, and catalase to neutralize ROS. Clinical relevance of oxidative phosphorylation. 1. Mitochondrial disorders. Defects in any ETC component can cause diseases such as Lee syndrome and MELIS, mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. 2. Ischemia reperfusion injury. During events like heart attacks, a lack of oxygen halts the ETC. When oxygen returns, a surge in ROS production can cause tissue damage. 3. Uncouplers. Compounds like DNP, dinitrophenol, or thermogenin in brown fat allow protons to flow back into the matrix without ATP synthesis, dissipating the proton gradient as heat. This can be beneficial for thermogenesis but dangerous if uncontrolled. Summary of oxidative phosphorylation. Purpose: to convert the energy stored in NADH and FADH2 into a usable form, ATP. Mechanism: electron transport generates a proton gradient which drives ATP synthesis. Yield: about 26 to 28 ATP molecules per glucose molecule through oxidative phosphorylation. Significance: it is the most efficient way to produce ATP, vital for cells with high energy demands like muscle and neuronal cells. Oxidative phosphorylation is a finely tuned process that integrates energy generation, cellular signaling, and adaptation to changes in oxygen availability, making it a cornerstone of aerobic metabolism. Glycogenesis is the process by which glucose molecules are polymerized to form glycogen, a highly branched polysaccharide that serves as a storage form of glucose in the body. This process occurs mainly in the liver and skeletal muscle cells, allowing the body to store glucose when blood sugar levels are high, e.g., after a meal, and to release it when glucose levels drop, e.g., during fasting or exercise. Overview of glycogenesis. Purpose: to store glucose in the form of glycogen for future energy needs. Location: cytoplasm of liver and skeletal muscle cells. Liver regulates blood glucose levels by releasing glucose from glycogen as needed. Muscle uses glycogen for energy during muscle contraction. Key substrate, glucose 6-phosphate, G6P, derived from glucose. Hormonal regulation, stimulated by insulin and inhibited by glucagon and epinephrine. Steps of glycogenesis. The synthesis of glycogen involves a series of enzymatic reactions that convert glucose into a polymeric structure. Below is a step-by-step breakdown. 1. Phosphorylation of glucose. Reaction. Glucose plus ATP, glucose 6-phosphate, G6P plus ADP. Enzyme. Hexokinase in muscle or glucokinase in liver. Importance. Phosphorylation traps glucose inside the cell and prepares it for glycogen synthesis or glycolysis. Number 2. Isomerization to glucose 1-phosphate, G1. Reaction. Glucose 6-phosphate, glucose 1-phosphate. Enzyme. Phosphoglucomutase. Importance. This reversible reaction shifts the phosphate group to the 1 position, which is necessary for glycogen synthesis. 3. Activation of glucose to UDP glucose. Reaction. Glucose 1-phosphate plus UTP, UDP glucose plus PPI, pyrophosphate. Enzyme, UDP glucose pyrophosphorylase. Importance. UDP glucose is the active form of glucose that acts as the immediate donor of glucose residues during glycogen synthesis. Pyrophosphate, PPI, is quickly hydrolyzed to 2 inorganic phosphates, driving the reaction forward and making it irreversible. 4. Initiation of glycogen synthesis. Reaction. UDP glucose plus glycogen primer, glycogen, N plus 1 plus UDP. Enzyme, glycogenin primer and glycogen synthase. Mechanism. Glycogenin acts as both a primer and an enzyme, starting the glycogen chain by attaching the first glucose residue to a tyrosine residue within the glycogenin protein itself. Once around 8 glucose residues are attached, glycogen synthase takes over to elongate the chain. 5. Elongation of the glycogen chain. Reaction. UDP glucose plus glycogen, glycogen, N plus 1 plus UDP. Enzyme. Glycogen synthase. Importance. Glycogen synthase is the main enzyme for adding glucose residues to the non-reducing ends of the growing glycogen chain through awful 4 glycosidic bonds. 6. Branching of the glycogen chain. Reaction. Transfers a segment of about 6 to 7 glucose residues from the end of a glycogen chain to a more internal position, forming a branch point. Enzyme. Branching enzyme, also called amylo-1416 transglycosylase. Mechanism. It breaks an AF-out-1,4 bond and forms an out-1,6 bond, creating a new branch. Importance. Branching increases solubility and accessibility of glycogen, making it easier to add or remove glucose units. It also enhances the rate of glucose release during glycogenolysis. Summary of glycogenesis reaction. Starting material glucose in the form of G6P. Intermediate UDP glucose. Products glycogen and UDP. Energetic cost, 1 ATP used by hexokinase glucokinase and 1 UTP used by UDP glucose pyrophosphorylase per glucose residue added to glycogen. Regulation of glycogenesis. Glycogenesis is highly regulated to ensure that glycogen synthesis occurs when glucose levels are high and stops when glucose is needed by the body. The main regulatory mechanisms involve hormonal control and allosteric regulation. 1. Hormonal regulation. Insulin stimulates glycogenesis in response to high blood glucose levels. Insulin activates protein phosphatase 1, PP1, which dephosphorylates and activates glycogen synthase. Insulin also inhibits glycogen phosphorylase, reducing glycogen breakdown. Glucagon in the liver and epinephrine in both liver and muscle inhibit glycogenesis during low blood glucose or stress. These hormones activate protein kinase A, pKa, which phosphorylates and inactivates glycogen synthase, thereby inhibiting glycogenesis. They also activate glycogen phosphorylase, promoting glycogen breakdown, glycogenolysis. 2. Allosteric regulation. Glucose 6-phosphate, G6P, acts as an allosteric activator of glycogen synthase, promoting glycogen synthesis when glucose is abundant. ATP, high levels signal a well-fed state and support glycogen synthesis. AMP indicates low energy levels and activates glycogen phosphorylase, inhibiting glycogen synthesis in favor of glycogen breakdown. 3. Regulation in muscle versus liver. Liver glycogenesis primarily responds to changes in blood glucose levels, regulated by insulin and glucagon. Muscle glycogenesis responds more to local energy needs. It is activated during periods of rest when G6P levels are high and inhibited during exercise when ATP demand is elevated. Clinical significance of glycogenesis, glycogen storage diseases, GSDs. Genetic disorders affecting enzymes in glycogen metabolism can lead to abnormal glycogen accumulation or breakdown, causing conditions such as von Gierke's disease, deficiency in glucose 6 phosphatase, or McArdle's disease, deficiency in muscle glycogen phosphorylase. Diabetes mellitus. In diabetes, the regulation of glycogen metabolism is impaired, leading to hyperglycemia and decreased glycogen storage. Hypoglycemia. In the case of fasting or excessive insulin, rapid glycogen depletion can lead to hypoglycemia, especially if glycogenolysis is impaired. Summary of glycogenesis. Glycogenesis allows the storage of excess glucose, converting it into glycogen for later use. It involves the activation of glucose to UDP glucose, its addition to a growing glycogen chain, and the formation of branches. It is highly regulated by hormonal and allosteric mechanisms, ensuring that glycogen synthesis occurs under conditions of glucose abundance and halts during glucose scarcity. Clinical relevance includes metabolic disorders that affect glycogen synthesis and breakdown, impacting the body's ability to manage energy reserves. Understanding glycogenesis is critical for appreciating how the body maintains glucose homeostasis and how it prepares for periods of fasting or increased energy demand. Glycogenolysis is the process by which glycogen is broken down into glucose or glucose 6 phosphate, G6P, to be used as an energy source, especially during periods of fasting or intense exercise. It occurs primarily in the liver and skeletal muscles, where glycogen serves as a stored form of glucose that can be rapidly mobilized when needed. Overview of glycogenolysis. Purpose: to release glucose from glycogen stores for energy production. Location: liver releases glucose into the bloodstream to maintain blood glucose levels. Muscle utilizes glucose 6 phosphate directly for energy within the muscle cells during physical activity. Key enzymes: glycogen phosphorylase, debranching enzyme, and phosphoglucomutase. Hormonal regulation, stimulated by glucagon and epinephrine and inhibited by insulin. Steps of glycogenolysis. The breakdown of glycogen into usable glucose involves a series of enzymatic reactions. Below is a detailed breakdown of the process. 1. Glycogen phosphorylase action. Reaction: glycogen N plus pi, glycogen N-1 plus glucose 1 phosphate, G1P. Enzyme: glycogen phosphorylases. Mechanism: glycogen phosphorylase cleaves Ha1-4 glycosidic bonds at the non-reducing ends of glycogen chains, releasing glucose 1-phosphate, G1P-STAN. Limitations: glycogen phosphorylase can only cleave glucose residues up to 4 residues away from a branch point, leaving behind a limit dextrin. 2. Debranching enzyme action. The debranching enzyme has 2 activities to handle the branches in glycogen. Transferase activity shifts a block of 3 glucose residues from the limit dextrin to a nearby non-reducing end, creating a longer chain that glycogen phosphorylase can continue to degrade. Glucosidase activity hydrolyzes the OC1-6 glycosidic bond at the branch point, releasing a free glucose molecule. Enzyme: debranching enzyme, also known as amylo-1,6 glucosidase. Result: allows the complete degradation of glycogen by making more glucose residues accessible to glycogen phosphorylase. 3. Conversion of glucose 1-phosphate, G1P, to glucose 6-phosphate, G6P. Reaction: glucose 1-phosphate, glucose 6-phosphate. Enzyme: phosphoglucomutase. Mechanism: phosphoglucomutase transfers a phosphate group between the 1 and 6 positions, forming glucose 6-phosphate, G6P. Importance: G6P is a key intermediate that can be used in different metabolic pathways depending on the tissue. In the muscle, G6P enters the glycolysis pathway directly to provide energy for muscle contraction. In the liver, G6P can be converted into free glucose and released into the bloodstream. 4. Conversion of glucose 6-phosphate to free glucose, liver-specific reaction. Glucose 6-phosphate plus H2O, glucose pi. Enzyme: glucose 6-phosphatase, present in the liver and kidneys, but absent in muscle. Importance: Glucose 6 phosphatase hydrolyzes G6P to free glucose, which can then be released into the bloodstream to maintain blood glucose levels during fasting or between meals. Summary of glycogenolysis reactions. Starting material, glycogen. Intermediate, glucose 1 phosphate G1P, glucose 6 phosphate G6P. Products, free glucose in the liver or glucose 6 phosphate used in glycolysis in muscles. Energetic yield. Glycogen breakdown does not require ATP input, making it an energy-efficient process for mobilizing glucose. Regulation of glycogenolysis. The regulation of glycogenolysis is tightly controlled by hormonal signals and allosteric regulators to ensure that glucose is released when needed for energy. Key regulatory mechanisms include 1. Hormonal regulation. Glucagon, liver, and epinephrine, liver and muscle. Glucagon, released during fasting or low blood glucose levels, primarily targeting liver cells to stimulate glycogenolysis. Epinephrine, released during stress or exercise, targeting both liver and muscle cells to increase glucose availability. Mechanism. Both hormones activate adenylyl cyclase, leading to the formation of cAMP, which activates protein kinase A, pKa. Result, pKa phosphorylates glycogen phosphorylase, activating it, and phosphorylates glycogen synthase, inhibiting it, promoting glycogen breakdown. Insulin, released when blood glucose levels are high, and it inhibits glycogenolysis. Mechanism, insulin activates protein phosphatase 1, PP1, which dephosphorylates glycogen phosphorylase, inactivating it, and activates glycogen synthase, promoting glycogen storage instead of breakdown. 2. Allosteric regulation. ATP. High levels of ATP inhibit glycogen phosphorylase, indicating sufficient energy availability and reducing glycogen breakdown. AMP acts as an allosteric activator of glycogen phosphorylase, especially in muscle, signaling low energy levels and increasing glycogen breakdown. Glucose 6 phosphate, G6P, inhibits glycogen phosphorylase and activates glycogen synthase, favoring glycogen synthesis over breakdown. Calcium, Ca-quamals. In muscle, Ca-mopin, released during muscle contraction, activates phosphorylase kinase, which in turn activates glycogen phosphorylase, linking muscle activity directly to glycogen breakdown. Differences between glycogenolysis in liver and muscle. Liver. The primary role is to maintain blood glucose levels during fasting or stress. Glucose 6 phosphatase allows the liver to convert G6P to free glucose and release it into the bloodstream. Responds to both glucagon for glucose regulation and epinephrine for stress response. Muscle. The primary role is to supply energy for muscle contraction. Lacks glucose 6 phosphatase, so G6P generated from glycogenolysis enters glycolysis directly to produce ATP locally. More responsive to epinephrine than glucagon, with regulation linked to local energy needs, e.g. AMP levels during exercise. Clinical significance of glycogenolysis. Glycogen storage diseases, GSDs. Disorders involving mutations in enzymes of glycogen metabolism can impair glycogenolysis and lead to conditions such as type 1, von Gierke's disease, deficiency in glucose 6 phosphatase, leading to severe hypoglycemia and glycogen accumulation in the liver. Type Phi, McArdle's disease, deficiency in muscle glycogen phosphorylase causing exercise intolerance due to impaired glycogen breakdown in muscle. Diabetes mellitus, impaired regulation of glycogenolysis due to insulin resistance or deficiency results in hyperglycemia. Hypoglycemia, dysfunctional glycogenolysis in the liver can lead to low blood glucose levels, especially during periods of fasting or after physical exertion. Summary of glycogenolysis. Glycogenolysis breaks down glycogen into glucose 1 phosphate, which is converted into G6P and then into free glucose in the liver. It is regulated by hormonal signals like glucagon and epinephrine and allosteric factors like AMP and ATP. It plays a crucial role in maintaining blood glucose homeostasis and providing energy during periods of increased demand. Glycogenolysis is an essential metabolic pathway that allows the body to quickly access stored energy and maintain glucose levels during periods when external glucose supply is limited. It complements glycogenesis and gluconeogenesis in managing the body's energy balance. Gluconeogenesis is a metabolic process by which glucose is synthesized from non-carbohydrate precursors. It is especially important during periods of fasting, starvation, or intense exercise when glucose levels are low and glycogen stores are depleted. The process ensures that tissues such as the brain, red blood cells, and renal medulla, which rely heavily on glucose, continue to have a supply. Overview of gluconeogenesis. Purpose: to produce glucose from non-carbohydrate sources, maintaining blood glucose levels during periods of fasting or starvation. Primarily in the liver, about 90% of gluconeogenesis. Kidneys also contribute, especially during prolonged fasting. Cytoplasm, mitochondria, and endoplasmic reticulum are involved in different steps of the pathway. Key precursors: lactate, from anaerobic glycolysis in muscles and red blood cells. Amino acids, especially alanine, from protein breakdown. Glycerol, from the breakdown of triglycerides in adipose tissue. Key enzymes: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, PFCK, fructose 1,6-bisphosphatase, glucose 6-phosphatase. Hormonal regulation, stimulated by glucagon, cortisol, and epinephrine, inhibited by insulin. Steps of gluconeogenesis. Gluconeogenesis largely reverses glycolysis, but because some steps in glycolysis are irreversible, it uses alternative enzymes to bypass these steps. Here is a detailed breakdown of the process. 1. Conversion of pyruvate to oxaloacetate. Reaction: pyruvate plus CO2 plus ATP, oxaloacetate ADP plus pi. Enzyme: pyruvate carboxylase, requires biotin as a cofactor. Location: mitochondria. Importance: pyruvate is carboxylated to oxaloacetate, which is a key intermediate in both gluconeogenesis and the TCA cycle. 2. Conversion of oxaloacetate to phosphoenol pyruvate action. Oxaloacetate plus GTP, phosphoenol pyruvate plus CO2 plus GDP. Enzyme: phosphoenol pyruvate carboxykinase, PEPCK. Location: can occur in the mitochondria or cytoplasm depending on the organism. Importance: this step bypasses the irreversible pyruvate kinase step of glycolysis. PEP is then converted to fructose 1,6-bisphosphate through several reversible glycolytic steps. 3. Conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Reaction: fructose 1,6-bisphosphate plus H2O, fructose 6-phosphate plus pi. Enzyme: fructose 1,6-bisphosphatase. Location: cytoplasm. Importance: this enzyme bypasses the irreversible phosphofructokinase 1, PFK1, step of glycolysis, allowing the synthesis of fructose 6-phosphate. 4. Conversion of fructose 6-phosphate to glucose 6-phosphate. Reaction: fructose 6-phosphate, glucose 6-phosphate. Enzyme: phosphoglucose isomerase, the same enzyme used in glycolysis. Location: cytoplasm. 5. Conversion of glucose 6-phosphate to glucose, liver specific. Reaction: glucose 6-phosphate plus H2O, glucose plus pi. Enzyme: glucose 6-phosphatase. Location: endoplasmic reticulum of liver and kidney cells. Importance: this step bypasses the irreversible hexokinase glucokinase step of glycolysis. Glucose 6-phosphatase is not present in muscle, so gluconeogenesis cannot release free glucose from muscle cells into the blood. Summary of gluconeogenesis reactions. Starting materials: pyruvate from lactate amino acids, glycerol from triglycerides. Key intermediates: oxaloacetate, phosphoenol pyruvate, fructose 1,6-bisphosphate, glucose 6-phosphate. Final product: glucose. Energetic cost: 6 high-energy phosphate bonds, 4 ATP, 2 GTP, and 2 NADH per glucose molecule synthesized. Regulation of gluconeogenesis. Gluconeogenesis is tightly regulated to ensure that glucose is produced when needed and conserved when energy is sufficient. It is regulated at several key points. 1. Hormonal regulation. Glucagon, secreted in response to low blood glucose levels. It promotes gluconeogenesis by increasing the transcription of pepke, a key enzyme in the pathway, activating protein kinase A, PKA, which inhibits glycolysis and favors gluconeogenesis by phosphorylating enzymes such as pyruvate kinase, inhibiting it. Cortisol, a stress hormone that enhances gluconeogenesis, especially during prolonged fasting or stress, by promoting protein breakdown in muscles, providing amino acids for gluconeogenesis, and increasing PEC expression. Insulin, inhibits gluconeogenesis by downregulating the transcription of peptide and other gluconeogenic enzymes. It also promotes glycolysis and glycogenesis, favoring glucose storage over production. 2. Allosteric regulation. Fructose 2,6-bisphosphate, F26BP, acts as an allosteric activator of PFK1, stimulating glycolysis, and an inhibitor of fructose 1,6-bisphosphatase, inhibiting gluconeogenesis. Levels of F26BP are controlled by insulin, which increases its levels, and glucagon, which decreases its levels. AMP. High levels of AMP signal low energy and inhibit fructose 1,6-bisphosphatase, reducing gluconeogenesis in favor of glycolysis. Acetyl-CoA, an allosteric activator of pyruvate carboxylase, signaling that there is enough acetyl-CoA for the TCA cycle and directing pyruvate toward gluconeogenesis rather than the TCA cycle. 3. Substrate availability. Availability of amino acids, glycerol, and lactate influences the rate of gluconeogenesis, as these precursors provide the building blocks for glucose synthesis. During fasting or protein-rich diets, amino acid breakdown increases, providing more substrates for gluconeogenesis. Gluconeogenesis versus glycolysis. Glycolysis is a catabolic pathway that breaks down glucose to produce ATP and pyruvate, while gluconeogenesis is an anabolic pathway that synthesizes glucose. Although both pathways share some reversible steps, they differ in their irreversible steps and use distinct enzymes to bypass these reactions, allowing the body to regulate both processes independently. Reciprocal regulation ensures that when glycolysis is active, gluconeogenesis is suppressed, and vice versa, to prevent a futile cycle where glucose would be synthesized and broken down simultaneously. Energetic cost of gluconeogenesis. ATP-TP consumption. Gluconeogenesis is energetically expensive, consuming 4 ATP from pyruvate carboxylase and phosphoglycerate kinase, 2 GTP from PEPK, 2 DH from the conversion of 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. This high energy requirement ensures that gluconeogenesis only occurs when energy is available and glucose production is necessary, e.g. during fasting. Clinical significance of gluconeogenesis. Hypoglycemia. Impaired gluconeogenesis can lead to dangerously low blood glucose levels, especially during fasting or in people with impaired glycogen breakdown. Diabetes mellitus. Excessive gluconeogenesis contributes to hyperglycemia in diabetes, as insulin fails to inhibit glucose production in the liver. Alcohol metabolism. High alcohol intake can inhibit gluconeogenesis because alcohol metabolism depletes NAD levels in the liver, limiting conversion of lactate to pyruvate and leading to alcohol-induced hypoglycemia. Gluconeogenic disorders. Defects in gluconeogenic enzymes, e.g. pyruvate oxalase or glucose 6-phosphatase, can result in metabolic disorders that affect glucose homeostasis, leading to conditions such as pyruvate carboxylase deficiency, results in an inability to convert pyruvate to oxaloacetate, leading to lactic acidosis and hypoglycemia. Type 1 glycogen storage disease, von Gierke's disease. A deficiency in glucose 6-phosphatase leads to the accumulation of glucose 6-phosphate and severe fasting hypoglycemia, as the liver cannot release glucose into the bloodstream. Summary of gluconeogenesis. Gluconeogenesis is crucial for maintaining blood glucose levels during fasting, providing energy to glucose-dependent tissues like the brain. It involves unique enzymes that bypass the irreversible steps of glycolysis, such as pyruvate carboxylase, PEPK, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. The process is tightly regulated by hormones, glucagon, insulin, cortisol, allosteric effectors, AMP, acetyl-CoA, and substrate availability to ensure glucose production aligns with the body's needs. Gluconeogenesis is a vital metabolic pathway that complements glycolysis and glycogenolysis, helping the body adapt to periods of low carbohydrate intake and maintain metabolic balance.